Welcome to our March Wildcatalyst seminar. My name is Chelsea DiGiuseppe. I'm the marketing manager for UNHInnovation. So this year our theme is Women in Research and Innovation, and so our aim is to explore opportunities, challenges, and pitfalls of launching or running a research and innovation-based startup, while also highlighting exemplary women in the field. So each seminar is led by an innovative female researcher or women who have founded or led research-based businesses. So before I introduce our speaker, I just want to say thank you to our generous sponsors. We have Hamilton Brooks Smith Reynolds, a Boston-based full-service intellectual property firm. Saunders and Silverstein, a law firm in Amesbury, Mass. That provides services related to trademarks, copyright, internet software, and entertainment law. Pierce Atwood, an award-winning multi-service regional law firm. And there's more information about them out on our sponsor table out there. So following today's presentation, we just welcome you all to stay and enjoy some networking, chat a little bit more with Kim, and enjoy the refreshments. Just want to remind everyone, if you're under 21, don't touch the beer. Oh, and if you're an I-2 passport student and you did not swipe in at the front, just see Heather over there, and she will help you out. All right, so our speaker this evening is Dr. Kim Brouwer. She has actually traveled all the way from the University of North Carolina at Chapel Hill, so I appreciate you taking the time to do that. Dr. Brouwer is currently the Associate Dean for Research and Graduate Education at UNC Chapel Hill's Eshelman School of Pharmacy. She's a distinguished professor with over 250 publications throughout her career. So while working at UNC Chapel Hill, Dr. Brouwer co-invented a drug discovery tool based on her international expertise in the field of drug transporters, and she subsequently co-founded a startup called Qualys Transporter Solutions and licensed that technology from the university in order to commercialize her work. Actually, our director, Mark Saddam, was the COO of Qualys, so that's the connection with Kim there. And so in this seminar, she's going to share her experiences and advice for founding and growing a research and innovation-based startup, and she'll also share her thoughts on the unique challenges that women scientists and founders face, and hopefully discuss her opinions on what resources are needed for the next generation of innovative females.
Oh, great.
Thank you.
Thank you so much for the introduction and the invitation to be here to share with you this story about the Qualys journey. And I heard something about challenges and pitfalls, and I'm sure that Mark immediately thought of me and the Qualys story. And as you can see this, is this one of these that, okay, this is trick. Oh. I guess it doesn't.
Yeah, actually, the laser pointer will not work. It gets swallowed up on the...
Okay. Oh, there's no mouse. Okay, well, how am I going to do this? I guess I won't point. That's okay.
Yeah, that should work. The walk up to it. This part should work.
Yeah, but that, you know.
Oh, the pointer?
Yeah, the pointer. Okay, well, he's... Anyway, yeah, it's been a long journey. And there were, as you noticed, 3 names to this company. So that gives you some clue that it wasn't the first time that things worked well. So just in the spirit of full disclosure, I did want to acknowledge that I am a co-inventor and co-founder of Qualys and the Beekler technology, that I do serve as a consultant for Merck, and also our lab is funded by NIH and has funding from Intercept, Otsuka, and also Gilead Sciences. So I like to start off with the bottom line here. So the lessons learned, there's quite a few of them. And we'll go through these as I go through the story. So I'm not going to read them for you now, other than maybe choose your investors wisely. That might be one of the take-home messages. And the other take-home message is that successfully, and however you define that, I guess, maybe acquisition of the company, successfully bringing research innovations to market requires a lot of time, effort, passion, perseverance, or persistence, and there's an element of luck involved, and you'll hear that in the story today. So I am one of these reluctant entrepreneurs. I am a scientist. I'm a clinician, actually, and my goal is to always think about the patient and what we can do to help patients. And the idea of developing technology that would allow us to increase or shorten the time of getting a drug to market, and to making sure that that drug doesn't cause adverse effects, specifically drug-induced liver injury, which I'll refer to every so often as DILI, drug-induced liver injury. And that's what my interest is. I never thought that I would ever tell a story like this in front of an audience like this, but I will say I kind of enjoyed putting this story together. I over the last, how long's it been, 18, 19 years, I have learned a lot, but rather reluctantly, I think, is the story for me. So when we take a medicine, everybody sometimes has different reactions to that medication. And variability in patient response to medicines is due to a number of things. One, the absorption, how the drug gets into the body, and where it distributes in the body. And transporters, which is the area of science that I work in, controls that in many cases for medications. How it's broken down or metabolized, and the liver is a key organ in metabolism. There's other organs, of course, but I'm pretty liver-centric. You'll figure that out as we go along here today. And then how it leaves the body, of course, that we can measure urine, we can measure feces, but another important route is via the bile here, from the liver to the bile, then into the intestine, and then reabsorption back into the circulation. And that was the fundamental focus of the technology I'll be sharing with you today. Every time that the molecule goes through the liver, there's a chance that it could do some damage to the tissue if it is a bad actor, if you will. And so understanding the toxicity, the potential toxicity or the liability of a molecule is something that drug development scientists have to always do in the process of getting the drug to market. And as you can see underlined, ADME, A-D-M-E, and T, ADMET, is the processes that we believe contribute in large part to variability in medications. So the original company name was ADME Tech. So the ADME Tech. So you get that right, clever, right? So as I mentioned, I come at this a little bit more from the clinical angle. And in my early days, actually in graduate school and as a postdoc, but when I first became a faculty member at UNC, I was involved in some industry-sponsored studies looking at site-specific absorption in the GI tract. And we developed a tube that where we could localize, even the pointer won't work there. So we could localize this tube into various regions of the intestine. Now the volunteer would swallow the top end there where you see the balloon, they swallow that end, not the one with all the cannulas on it. And a good gastroenterologist could localize that into the stomach, through the stomach, into the duodenal region, or the jejunum, or the ileum, or even into the colon. And we would administer drug through the tube to various sites, and then look at where the compound of interest was absorbed. And so this tube we developed by putting a balloon on the end, molding together some tubing so that we could suction samples out. And I got to thinking that if we inflated this balloon, we could actually collect bile as it's dumped into the intestine. And so this was just showing the localization. It's a little bit hard to see without a pointer, but the tungsten tip, which is on the end below the balloon, is at point C. You can see it winding around the intestine there. And the clips that hold the balloon are at points A and B, if you can see that. Anyone want to volunteer? We're always looking for volunteers for these studies.
Can you bring one along? Grad students are for it.
A good gastroenterologist can get you to swallow anything. It's pretty amazing. So we developed this protocol in healthy subjects where We would feed them fatty meal to maintain gallbladder contractility. Then we would localize the tube. We'd give them an IV dose of this imaging agent. I'm going to show you in a video a minute of what this looks like when we use gamma scintigraphy to measure over the liver region. And then at a certain point in time, we can IV administer cholecystokinin, which is a naturally occurring peptide that causes that gallbladder to contract. And you'll see the bile pulsed into the intestine. And if we have the balloon inflated, and these little ports here show the holes that we had, we can actually suction the contents from the bile that's in the lumen of the intestine. We can suction that out through their mouth and everybody's happy, right? Yeah. So that was, but UNC wasn't interested in doing anything with this technology.
Has a limited market.
Okay, but we could measure biliary excretion in humans. And so here's the video. You know, how is this gonna work? Let's see, maybe if I just advance it. So this is uptake into the liver, very rapid. Within minutes you can see it. It's collecting in the gallbladder here. See a couple of spontaneous contractions. Then we administer cholecystokinin, and you'll see all of a sudden all of this being suctioned out. See there, it's going through the intestine and we're collecting it all. So it was really a pretty clever method, yeah. So we could measure biliary excretion in humans. But of course, we can't do this every day. Nobody, you know, how many people are really gonna want us doing this kind of a study, right? So we needed to think of a way that we could measure biliary excretion in humans, or at least predict it. And we actually had a method that we could use in some limited studies to quantify it. So we had been working with hepatocytes in our laboratory and went to a conference, and what we learned is by looking at a poster from Ed Lecluse, who was a co-inventor of this technology, is that if you take hepatocytes that you've isolated, and you can use your species of choice, so it can be rat or dog or mouse or human, and take those liver cells and seed them on a collagen-coated plate, is what we used in the beginning. Now we use BioCoat plates. And then you overlay them with a collagen or Matrigel or something, because hepatocytes like to be next to each other. They're very social, and they like to have something above and below them, because that's how they are in your liver. And over days in culture, these cells, they get together and form these bile ducts in vitro, bile ducts. And you know, I always said I'm not very smart. I don't really have any special talent. I'm just curious, and I'm always asking questions. So at the poster, I kept asking Ed, so what are those white belts that surround the cell? What are these things? And do they really collect stuff that the hepatocytes have secreted, like bile contents? And he assured me that they did. And of course, in those days, the NIH study sections, I have some critiques that say that there aren't even any transporters involved in drug disposition. So why would we think that this would even be a useful technology? But they were wrong. So what we did was we took these sandwich cultured, and we started with rat hepatocytes. And we looked at different proteins that we know, transport proteins that we know are important in biliary excretion of drugs, like MRP2. And over days in culture, we could see the expression of the protein. And when we did immunohistochemical staining for MRP2, which is in green, you see this chicken wire appearance, like what we would expect to see with the bile canalicular network if you were to slice through a liver. And in red is the MRP3, which is a basolateral protein. So MRP2 is on the apical or canalicular domain, and MRP3, shown here, is on the basolateral membrane. So you notice those don't co-localize, they're on different membranes. So we were really excited by that. And then we administered to the hepatocytes in culture on day 1 this carboxydichlorofluorocene diacetate. And as the diacetate, it passively diffuses into the cell. The esterases cleave it into a fluorescent carboxydichlorofluorocene, green. Well, here it shows white. And you can see that on day 1, there's mostly just accumulation in the cell. But look what happens at day 4 when we administer the CDF the carboxydichlorofluorocene, it localizes in these canalicular networks. We were really excited because we knew that if we could then figure out a way to measure that, we could quantify biliary excretion in vitro. So we did some electron micrographs, and we saw, in fact, these junctional complexes, the tight junctions, that would form this bile pocket, or this canalicular pocket, I think is the best way to describe it. And you could see the microvilli extending in. So this confirmed that, in fact, that was a mechanism by which we would be able to accumulate the substrate. And so knowing that tight junctions are sealed with calcium, we realized that if we incubated the well with a standard buffer containing calcium, we could measure accumulation in the cell and the bile compartment. And in a side-by-side well, if we just incubated in a calcium-free medium, that would disrupt the tight junctions. What was in the bile network would leak back into the medium, and there we would just quantify what was in the cell. So measuring substrate in cell plus bile, and in another well, substrate in cell, and taking the difference, not rocket science, you don't need any high-powered math, just subtraction, you could quantify the substrate in the bile compartment. And well, this just shows accumulation in black bar cell plus bile and open bar cell to give you some idea. With bile acid tarcholate, you see a pretty extensive biliary excretion index, which we calculate just by the accumulation in cell plus bile minus accumulation in cell normalized for what's taken up. And then, because I'm a pharmacokineticist and we know that the percent or the mass isn't really what we would want to scale from in vitro to in vivo, it's actually the clearance term that you would scale, we devised an equation to estimate the biliary clearance. And then we could correlate that with our in vivo data. So we had lots of rat data where we could compare our in vitro estimated biliary clearance to our in vivo measured biliary clearance, because rats don't care if we cannulate their bile ducts, and we can easily quantify what comes out in bile. But in humans, the green, that was the blue data points. But the green data points, you see, we only have a few compounds because running those clinical studies wasn't so trivial, and we were somewhat limited in what we could do. So that was the essence of the technology. So what did I learn? I learned, I was just a young assistant professor. I didn't know anything about what you should and shouldn't publish, and when you should, if you had some kind of a clever idea, when you should or shouldn't submit an abstract. I had a graduate student, Shenrong Liu, that was heavily involved in developing this. So, and we were NIH funded. So of course, we've got to present our work at meetings and publish. And so the challenge here is timing and method. So this is a method. This isn't a molecule that we're going to develop into a drug. It's a method. And once you describe your method in your publications, anybody can do it. Or at least that's what you hope, right? That's what NIH certainly expects these days, although they're finding that that's a separate issue. So how do you protect your technology? And that has been, wouldn't you say, Mark, one of the biggest challenges with this technology?
And actually, can I jump in for a second?
Oh, please.
So for those of you who don't know, you introduced it. So I was employee 4 in Qualist, and so this was what we had. So where we're stopped here is where we kind of, well, you'll go through the story, but where we spun the company at. So we had a methods patent, and if you could go back to the last slide, so basically the pharmaceutical industry, when it came to commercializing it, looked at this and were like, it's a method, how are you going to tell us? And so there are two things on this, well, there's one thing on this slide and one thing on the other slide that wound up being the story. So this biliary clearance index is, you can't get that without using the method. There's no other way to calculate biliary clearance without using this method. And so it was actually the calculation, the claim that said this is the calculation, that was what we were able to build the company around. Because people would say, oh, the biliary clearance of this drug in vitro in the cells is this. And we'd say, well, you have to be using our stuff because the only way to get it is to use this. They'd say, no, we didn't. They're like, well, show us. They're like, we don't want to. So even though it is a methods patent, the fact that we effectively created a proprietary calculation, and that calculation was what was meaningful. Then on the next slide, and I remember doing this because I was out there trying to sell this stuff, was those 3 green dots. One more n than, or the last n that you need to make some conclusion, an n of 3 versus an n of 2. So we could actually say, this is so the rat correlation was really pretty good. It was like 0.98 or 0.99 or something like that. But we said, and there's 3 human dots on this, and this is real human, so the story of the canaliculi and the video and stuff wound up being, I could say to people, 3 humans did this. This is human data with this cellular data. So this is what happens in people. And even with just 3 people, that was enough for people to say, okay, we'll give it a try. If we didn't have those 3, if we didn't have that great video, to me it was a great video because it was like $3 million video with what we made off of it. But if we didn't have those 3 data points, no one in science would have believed that this would transfer from rat to human, because in your scientific presentation you showed earlier today, the way that rats metabolize things and clear things is fundamentally different than humans. In fact, it almost looks, in some level, at least at the bile acids, it looks nothing alike. So we were always stuck in this model of, well, yeah, it works in a rat, but it doesn't work in a human. And you could just go around in that circle forever if we didn't have those 3 data points. And so effectively, the crux of what the company was able to do eventually are those 3 dots. So an N of 2 is no good, but an N of 3 sometimes can be pretty meaningful. So the third one's at the bottom.
Well,
it's at
it's
0.
down here at the bottom.
It counts.
So, this is kind of interesting. I hadn't planned to digress to this, but over the last 5 or so years, there has been an unbelievable amount of work on correlating in vitro to in vivo. As you can imagine, there's a lot of power in that. And in the drug development, the focus now is to use scaling from measuring the protein by mass spec from the in vitro data, and then putting that into equations to scale to human. And I'm amazed at the effort that has gone into way beyond what I ever would have thought about in quantifying the protein to get this biliary clearance in vitro calculation to then scale to human in vivo. So it's been an interesting ride. Anyway, so I guess there's some things to think about in methods, and just being careful that you don't disclose things too early, because I'm sure all of you who've been much more trained in the do's and don'ts of developing a technology knew, but just out of ignorance, we made a few mistakes there. So at the same time we were working on this technology, there were 2 other faculty at UNC that were working on other ADME technologies. And at that time in the industry, so this was in the late 90s, there was really just the realization of how important ADME was in drug disposition. What I described to you in that first slide, which now seems so obvious, was definitely not that obvious back in the 90s. And developing methods to do higher throughput screening was in its infancy. So certainly nowadays, I mean, that's common in drug development, and there's lots of screens available. But this was one of the foundations on which the company was developed. And so the beginning of the I guess the idea of a startup was really focused on revolutionizing the ADMET technology, the disclosure, I guess, in '99 of this technology. Darren Thacker, who was a co-founder of the company, is currently our interim dean in the School of Pharmacy at UNC, also had some technologies, went to our Office of Technology Development and really convinced them that that we should file patents, put forward patent applications and move forward with a spin-off. And Darren was really the leader here. And all I can say is, if you're a scientist and kind of a reluctant entrepreneur like me, you need somebody that's willing to invest the time and effort. Because at that time in my career, and Mark can attest to this, to me it was somewhat of a distraction, all of the the buzz about being an entrepreneur. Darren's technology, he was doing some work in the area of absorption enhancers and also had a metabolic stability screen. Gary Pollock, a longtime colleague of mine at UNC, had a very clever high throughput protein binding assay, AcuPro, which I think that there's still a lot of interest from the industry in. So that's not a dead technology yet. And then, of course, the B-Clear. And I do want to attribute Ed Lecluse as a co-inventor and Shinrong Liu, the graduate student, who none of this would have been possible without them. So here's the timeline. In 1999, Darren was really leading the charge for the company. This guy sitting over here was at our OTD and involved. We needed money, and I had a NIH grant. I guess at that time I had an R01, but it wasn't much to do much with. These clinical studies were expensive, and we didn't have the resources to look at all the different matrices and the overlays and all the things that needed to be done. So Pfizer offered to set up a contract with us. And what was it, 100,000, I think, or something? I don't know.
It was 500,000 over 3 years. It was more than you think.
Well, it wasn't nearly enough. And in exchange, they got the license to use the technology forever. It was a bad deal. But at the time, it was all we had, so we went with it. To this day, Pfizer still uses this technology a lot. Now, it's always been a collegial relationship. I hope there's no one from Pfizer in the room. And they have helped develop the technology, and they've published and done really good science with it, and I think we've learned from each other. So maybe it was okay. But another take-home message here is just be careful who you make agreements with. At the time, it might seem like a really great opportunity.
Well, and what I will say is that, and I have the mic on so they can hear me, is that the fact that Pfizer was publishing on the work was industrial validation of the work. So as soon as they started saying Pfizer, a name from Pfizer was on a publication with you, the industry said, oh, well, this must be interesting because Pfizer's publishing on it. So it was the first sort of proof of concept in the field was the Pfizer work and the BMS work that eventually got published. And that's where it started to look interesting to other people because Pfizer was and still is the largest pharmaceutical company in the world.
That's right, well respected. And the fact that they could reproduce the technology was really reassuring to me, as you might imagine. There was an investigator from Europe that had claimed that the technique was not reproducible. In retrospect, I think the problem was that they didn't have good enough quality hepatocytes. The starting material is really important, and we didn't realize that at the time. So there's, when you have a method that may have some unique features and the starting material is really critical, I think it's good to know the limitations of your system always. So, I forgot. In 2001, Doug Mendenhall accepted the CEO position and Paul Mafouad the CSO position. They were both former Glaxo GSK employees, and there was interest among VCs. I think you and Darren were out on the roadshow, selling the technology, a lot going on, and then bad timing. So again, the luck element here.
Well, and the one slide that I couldn't reproduce. So we were actually waiting for a term sheet from a venture capitalist on September 10th. We expected to receive a term sheet on September 10th from a local VC in the area of $3 million, and it didn't show up. Not because of 9/11. That was just didn't show up. And by 10 a.m. the next day, we were done. There was not, you turned on the TV and said, this isn't happening.
Right.
So.
Timing. So in that unfortunate time, really, we did hire Hutchinson & Mason as corporate counsel. They were very helpful. OTD founder partnership at UNC did kind of keep things afloat. The school was particularly strong in providing support during that time, and I'm grateful to the support at UNC OTD for their help because there were patent costs that the university had incurred, and so that's problems. So we were sort of floundering around. The CEO and CSO left. We regrouped to search for a CEO, had some ins and outs. This is maybe the typical story, right? We did find a CEO, Scott Neville, and at that time the co-founder, Ed Lecluse, left. And then things started to turn around. We got the licensing agreement. Some angel investors appeared on the scene. Qualist was launched in 2003. Now, and previously we had called this company ADMET. I forgot to mention that. But when the company was launched, they wanted to change the name, so they changed it to Qualist Transporter Science Transforming Results. And everybody was excited. Scott, the CEO, did have some life science experience. We did have a CSO come in, Ken Brouwer, happened to be my husband. That was convenient. That worked out really well. Ken had quite a bit of experience, 18 years at Glaxo, and had the background in drug metabolism and kinetics and transport, everything we needed. And the focus of the company, of this Qualist, was really on accelerating drug discovery and development. And this was the Qualist slide that went on the roadshow, and there were a lot of them to really try to move things along. So in 2003, at that time, the company had 8 full-time employees. There was a strong IP position, productive R&D, an active advisory board. And one of the things that the advisory board recommended was that because there were 3 technologies, the company was not very focused. And they advised that the company focus in on the technology that had competitive advantage. And that, I don't know, maybe Mark, you have a different perspective on this, but I think that the unique feature of the B-Clear assay, the biliary excretion, was that it was the only method that's available to quantify biliary excretion in vitro.
Yeah. We used to say it was the E in ADME. So that was, if ADME or ADMET is the field, there was no E, and we filled in the E.
Mm-hmm. Yeah, that's a good analogy there.
Market.
Yeah. I never took those classes. I'm a pharmaceutical scientist. There was, I think the business strategy at that time was the CEO's strategy was to license the technology, but it was pretty expensive. And so that didn't go quite as well as had been hoped. But what was going really well was contract research services, because it turned out that this technology took some expertise. So you couldn't just sell this to have just Joe Blow at company Z go license the technology and then use it and without some significant training and expertise. So the company got the bright idea that they could sell these B-Clear kits to simplify things, and that was a really, I think, a wonderful idea. Then in 2007, the CEO left, and Mark was the COO, and the angel investor became the acting CEO. Unfortunately, the angel investor seemed to just be sucking the lifeblood out of this company. I have nothing good to say about this angel investor. I will never utter his name publicly. He deserves no recognition. I'm sorry to say. He really, there wasn't really a sales force. I think Mark and Ken were out trying to sell this technology that just wasn't trivial. They were struggling to make payroll. It was a really bad situation, and I thought, this is probably the end of the road for sure after so many years. And whoops.
This is the one I added.
Oh, you added
this. I'm going to jump in here.
So maybe you'll have some good news.
No, there's no good news at all. So a couple of things. So this the CEO leaving was not willing. The CEO is escorted to his car by the investors. And the story, I don't know if you've ever heard this, but the story was, so I'm sitting at my desk and I have a window behind me and I'm typing and I see something move and I go, oh, interesting. Our investors are here. Usually I knew. And I didn't know they were coming. So I said, oh, that's great. So click, 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 click. And then I see Mr. Neville walking to his car. I'm like, oh, well, he must have forgotten something in his car. And then I was like, that's unusual. And then the rear headlights went on, or the taillights went on, and then he pulled away. And I went, oh God, he's been fired. So I was watching the business execution of the CEO in my window and had no idea what was going to happen. So within 10 minutes, he who shall not be named, had called us in and said, he's been fired. We don't know what's going to happen. In fact, they said they were, at that time, even in 2007, they said they were probably going to wind the company down. And it was, what are we going to do? And so we figured out we could probably sell enough in 90 days to pay people, and I convinced the investors to give us 90 days of money so that we could try to solve the problem that was kind of obvious. And so even this financial struggles was basically the entirety of the timeline from 2007 until 2011. The other thing that, before I get to the other slide, is, where was it? Oh yeah, the licensing. So you talked about the patent costs. So this is one of the things that, I'm going to say because it was my decision. At the time we signed the license agreement, and by the way, for UNH faculty, we'll never do this, so don't ask me, don't say you did it before. We had a $250,000 patent fee due. So the university had kept the patents alive to the tune of $250,000 because honestly, I believed it was going to make it. I don't know that I told my boss how much we were actually spending, and since he didn't ask, I just kept, approving the patent bills. But had the university not held on to those patent costs for that long, nothing would have ever happened because we would have just abandoned them, and then there wouldn't have been anything to protect. So that's the luck part was, they weren't paying attention to the bills, I guess. So the other part,
the accounting system was so bad.
At UNH. We pay attention to the bills, so that'll never happen. So what I added just this one slide to sort of color commentary. So we started the company in 2003 with the model of licensing. That was what Scott set out to do. We were licensing to pharma, we train them on the techniques, and we do yearly license fees. And the license fees were the tune of $100,000 to $200,000 a year. Now, why did we do that? Now that seems ridiculous, but there were other technologies in the preclinical in vitro space that had used this model. And so it was a successful business model. We thought, oh, well, we'll do the same thing. Well, in the 6 months it took to launch the company and then figure out what we were going to do and go on the road, the entire pharmaceutical industry decided they were never doing that again. So the fundamental business model that we were using to launch the company and raise $3.2 million was dead in 6 months. So we had to figure out what else could we do since they didn't like anything that we had to say. So the second strategy was to do a services bit. So we started doing the services. And because like what Kim said, it's a really heavy, it's a precise technique. It's not like you need $4 million of equipment, but you had to be good at what you were doing. And so companies were saying it doesn't work, we tried it, which is a euphemism for we're infringing your patents and we don't think it works. And so we'd say, well, we need to try it for you. So we convinced them to do these service projects. What that required us to do at the university was we had to convince them to rewrite the license agreement because the license agreement was about the sub-licensing model in Strategy 1. So now the interesting part is that since I was in the tech transfer office and licensed the technology to ADMET, then I left and joined the company. I got to negotiate against my own license agreement. And so the fun part was at least the first 2 times when they said, well, the license agreement says this, and I would say, well, that's not what I meant when I wrote it. And they were like, okay, you get that twice. And so the first time I got it, and the second time they said, that's the last time you're ever going to say that. So when we went to services, we actually had to change what the definition of net sales was, how the university was going to get paid, when they were going to get paid, and so that kind of worked. So we had to rewrite the license. Then in 2007, services is really hard because basically, at least in the pharmaceutical industry, they don't believe you until they do the work. So like you said, it was Pfizer could reproduce the work. So it was only when they could reproduce your work that they said, oh, this must work because we can do it. So we're selling services and they'll say, all right, well, we paid you $50,000 to do the service, but we didn't do it. So how do we know you're not lying to us? That's not really what they say, but it's kind of what they mean. And so we said, well, we'll have to find a way to get them to do the work so that they believe that the data is useful. So it's this like, we're just spinning in circles. And so the third strategy was that products sell services. So we created the Be Clear plates, the hepatocyte plates, and we said, well, we'll create research tools that will sell to the pharmaceutical industry. They'll use the tools, and then they'll understand that the technology works, and then they'll hire us to do the services. So that was fine, except we had to rewrite the license agreement again, because now the business model was different, and how we got paid was different, and where it happened to different. So the good news was they realized what a pain in the butt it was to do the technique, so they were really interested in buying the services. And that was in 2007. That was when Scott was escorted out the door. And then in 2009, it actually flipped. It was the services actually sold the products. And so we thought that the service or the products were the Trojan horse to sell the services, and it turned out to be the other way around. So once they got used to having the products, and they said, oh, well, we can do this ourselves, then we don't need you. And so it actually flipped again. So we had to rewrite the license agreement again, because so I effectively rewrote my own license agreement 4 times. So the fourth time was when they said this doesn't matter anymore. So so effectively the companies wanted the products, and some of it was because this is now 10 years on. So the technology, you started publishing, there's stuff out there, there's stuff out there with Pfizer and Bristol Myers and Gilead Sciences and Array, Biopharma, and so the pharmaceutical industry is publishing saying this is good. So then it was easier to sell the products because they're seeing all of these publications from their peers who are publishing it, which means they kind of believe the work. And that model then, the products eventually was what got the company to sustainability after the new investors came in, which you can talk about.
So while these guys were having all this
and then
fun,
I quit and I came here. So the rest of it, because of who shall not be named.
They're having so much fun with this company. I was trying to just do science, remember? Clinician scientist. So in addition to predicting biliary excretion, my lab was looking at knocking down or knocking out particular proteins to understand what molecules in drug development might be using, which transporters to get into and out of the hepatocyte. We were studying, this was what the science discussion earlier today was about, the importance of route of excretion. The hepatocyte, it can excrete into bile, or it can excrete back into the blood, and then those compounds go out in the urine. And we could assess that. This is still a huge issue in the industry, how you measure intercellular concentrations. Well, in the sandwich cultured hepatocyte system, you had the cells right there, and all you had to do was sample or take out the cytosol, isolate it, and measure the unbound drug or metabolite concentration. And also looking at different interactions. So we had all of these potential further developments of the technology that could have been taken advantage of, but the angel investor that was basically in charge was just, there was no money to do anything or sell anything. So I don't need to say too much, that this was kind of where we were. But times changed. We had an opportunity for a new beginning. I'm not exactly sure how that all transpired, but a new set of investors appeared on the scene. Marcus Kroll and his colleague Heinz from Switzerland. They are not scientists, but they truly wanted to understand the technology, and I really think that they had a passion for developing tools that could be useful in drug development. Very thoughtful individuals. A new CEO, Chris Black, who was a scientist who understood the hepatocytes really well, and new company name, of course, so this is the round 3 here. The investors realized the importance of selling this technology, so a great sales force, and they had a strategy. And this change occurred in 2012, and within a year they began to sell transporter-certified human hepatocytes. So only about 50% of cryopreserved hepatocytes have transporter function. And if you don't have transporter certified hepatocytes, you won't be successful in measuring biliary clearance or studying transporter function. So this was, I think, really a clever business strategy. And then went on, most recently, they've developed this C-DILI assay, which is a way to measure in vitro using the sandwich cultured system the likelihood of a molecule causing drug-induced liver injury in vitro. So I think things really going well. I hope you can read this. So the Salesforce story, again, being a scientist, I never really appreciated the importance of being able to sell something, but it is really important. But those salespeople have to have the right qualifications. And that, in this particular case, I think was very important for the technology. So hopefully you get the message here with the Salesforce. And the company, I guess in 2017, ironically, the announcement came out on 9/11/2017. The company was acquired by BioIVT. Here's Marcus, again, not a scientist, but holding up one of the posters that we used to, Qualist would print their science for posters at meetings on the fabric, and Ken, the chief scientific officer, he was congratulating him, giving him a poster as the company was exiting, and Marcus liked it so well that he made a scarf out of it, and he was really a great CEO. I mean, I just can't say enough positive things about the turnaround that the company had with the investors. And you know, the acquisition in 2017, I think that, I guess that's what's defined as success for a spin-off company. I will say that there's still more lessons to be learned with this. Their sales, a lot of the sales force for Qualist left, and the BioIVT was a tissue reclamation company, and selling products, cells and blood and stuff is really different than selling science. So the Salesforce cartoon kind of is applicable even now. But anyway, so you know, my take-home messages, and I don't think this is gender specific. So I think that, to be honest, and I'm an academic scientist, are poorly prepared. Now things are getting better. I think at least here it's obvious, and also at UNC there's lots of opportunities, kickstart, and mechanisms for learning about and how things to do and not do to avoid all the pitfalls, and there's been plenty of them in this story. There's no question that the founders have to be totally committed to the science and believing that the technology is really, there's a lot of potential in that. That's absolutely critical. There needs to be seed money and incubator space. I think otherwise the inventors, the founders are really going to struggle and end up maybe in this is a great example of what not to do. And then clearly it's about people, making sure that you've got the right people at the right time doing what you need them to do to help the company be successful. And it just takes a long time. So there's lots of people to thank in this story. The co-founders, as I've mentioned, of course, Ken and Mark. I think they should win the prize for making the company successful in some very difficult times. Marcus, the last investor, and Heinz, his colleague, and also UNC, our Bill Campbell in particular, the dean at the time in the early 2000s. And the Qualist Advisory Board, some really high-profile scientists, Yuichi Sugiyama, Ron Borchardt, Anthony Liu, and Richard Kim, I particularly want to thank, and all of you who pay your taxes to support the research that was behind Qualist Transporter Solutions, and the very beginning ADMET. And and my students who had to put up with this periodically, all of the issues, students, postdocs, visiting scholars, including the Paul Watkins and the DILISIM team that I didn't talk about in much detail. And I'd be happy to answer any questions, or we can have some good discussion. Thank you.
So I'll ask the first one, which is rolling back to, I know the answer, but I'm going to make you answer it anyway. So going back to 1998 and 1999 as a associate professor, and the concept of the startup came up and Diren wanted to do it. What did you think? What did you think about doing a startup?
Don't bother me. I think, well, I'm a very practical person, and I'll have to say that being an entrepreneur never really wasn't what I dreamed of. I guess I dreamed of doing things that would make a difference in patients' lives, but the whole process, and even it was very interesting to look back over this story and everything learned, but would I do it again? I'd do it differently for sure. I think I would have probably been a little more involved.
But so then part two is, so having done it and seen your science in the market and doing things, so how did it flow back into your lab? So you've worked with, the top 10, 20, 30 pharmaceutical companies. You see your science being useful to them. How does working with industry, like you have some industry funding, but what did that do to your lab? How did it change your perspective as you were, I would qualify Kim as one of the purest academics that I ever worked with. You were the academic's academic. That's all you wanted to do. And this was all noise. Thankfully, you married somebody who wanted to do this kind of stuff. But now 20 years later, you work with industry a lot. How does that change how you look at your basic science?
Well, the industry funding, I think, and collaborating with industry is really helpful because it gives you the real world perspective on how your science is being applied. And that can be really critical in the types of questions that you decide to focus on and answer, and really what you do with it. And I think that there's just been a big cultural shift towards more of a focus on innovation than there was in the 90s and even early 2000s. And at UNC now in the School of Pharmacy, we have an Eshelman Institute for Innovation, and we aspire to have a culture of innovation at all levels, at student, staff, faculty, and everybody can invent things that, even just day-to-day things that you do, think creatively about how we could do things better, more efficiently. And so really inspiring that in graduate students and postdocs in particular, I think as part of the experience I've had with Qualist has really helped me change my opinion about the importance of being innovative and creative. But the business side, I knew I was not well trained in it or trained at all in, and so for me that was the aspect that there just wasn't enough hours in the day to develop expertise in that and stay on the cutting edge of science. So I think if you're a scientist, I think you have to make some hard choices because you can't do everything. And to be honest, maintaining NIH funding, our lab has been funded since '91. I have funding through 2022. Continuous funding has been challenging, and training graduate students and postdocs to go primarily to the industry to be at the cutting edge of science to improve drug development. That was my primary focus. So. Other questions about?
At what point did you realize that you had something that you could market, like yourself? Was it, because it's kind of fuzzy when you started getting involved with industry, with Pfizer. Was it when you were involved with Pfizer already?
I think when we signed that contract, and actually in looking back on it, I think the scientist from Europe that was not able to reproduce the technology and published that. I think at that point I realized that we had something that would be useful, and it was clear that others, industry does things that they don't talk about, so you don't really know what's going on behind the scenes, but there were a lot of companies that were looking at this method, and a lot of companies are to this day using the information without any sort of compensation. But in a way, that's satisfying because I realize that that's helping them select better compounds to take into development. So I guess it depends on, are you there for the financial gain, or are you there to really, help a patient in the end?
Well, after being at the scientific presentation, I was just wondering.
No, it was probably in the early 2000s. It wasn't immediately that, I think biliary excretion, how important is that? But it's still to this day for some drugs. There was just, when was the tolvaptan-Dilly case? I mean, drug-induced liver injury is fatal in some cases. And when a drug gets to market and then you have like the triglycerin story, sort of the classic example where you have a number of fatalities after the drug has been on the market. I mean, that is a huge issue. And so if we can prevent those by using this kind of technology, then that's what really matters. But I didn't realize that in the beginning. So it was probably in the early 2000s. So you had a question too?
Is there a marked way that it's changed, the trajectory of your science, or you kind of feel like, I was going on this path, I'm still going to be on the path, it doesn't matter what happens, I'm on this path. Did it change like your ability to get funding when you were writing those future NIH proposals? Or?
It's definitely impacted the direction of the research, absolutely. In the early days, before we really just developed the B-Clear system, I was primarily looking at site-specific absorption of drugs in the intestine, as I described. So and so that was one important area, and transporters, people didn't even realize the importance of those. So some of this, we were about 10 years ahead, I would say, of the technology in '99. Most scientists in the industry or elsewhere didn't appreciate the value of that technology because people didn't realize the importance of drug transporters at that time. Today, people realize that. But
and am I right in this story that it all came down to like you were standing at this guy's poster? Like, what is that thing?
It really was. So I always tell my students to pay attention when they go to meetings and when they hear people talk and to go through the posters, because it wasn't something that we were actually looking for. We were using some different methods at the time to try to quantify biliary excretion, and I was so frustrated with those methods that it was sort of a desperation of there's got to be a better way to do this science. So yeah, it was really that. Mm-hmm.
We probably have time for one more question.
And then it's time for beer, right?
Yeah.
Well, I'm going to ask. Does anyone else have one? Because I want to go back to the gender thing because you didn't really cover it, but I want to ask the question as a woman in science pushing against convention, you're pushing into new areas of science. So, do you have any stories, or can you think of cases where that came up in terms of trying to do this and trying to drive what is effectively a new area of science, at least the transporters and the value transporters in the beginning? Is there any point where you felt like being a woman, you were at a disadvantage, or you felt like that has come up at any point?
I don't think so. I'll have to say maybe I was just so focused that I didn't pay attention to what other people might have been saying or thinking, but no, I can't really see how I was treated any differently.
The answer can be no. That's fine.
So I think that some of that is the culture at UNC, although the culture has changed a lot in the 20 years that this technology was being developed. But I can't really say that related to Qualist I ever felt any time. The patent when we went to the patent office to
interview.
The interview. It happened to be a woman, so maybe that was a plus, right? And she was very kind, I thought, and she was a scientist, asked good questions. We had a good discussion. So, but I don't think there was anything special about that. So. Oh yeah, that angel investor. The day I see him. Yeah, the investors were men. Yeah, but I was pretty separated from all of that. Yeah. And I think, another thing, I guess certainly Ken had to deal with a lot of the issues with the company, and so I think we were fortunate in that respect that we could that it worked out well, and it was kind of a dinner conversation. How are the hepatocytes doing today? What ideas? But no, I think I think you just have to be you have to be persistent. It doesn't matter who you are. If you're a scientist and love science or whatever you do, you've got to have passion for it, and that's gender independent. So thank you very much. Good discussion.